Good afternoon, everyone. Good afternoon. I hope you're having a great day here at our 11th annual Undergraduate Research Day. Really welcome you to York College. For those of you who are visiting us for the first time, we really appreciate you being here. I am here simply to introduce our wonderful grants officer who is behind the scenes, the backbone of all of this, helping our faculty get grants. And just for the York folks who are in the audience, we do PSC CUNY grant writing boot camp every fall semester. So if you're looking for money for travel or whatnot, please look for the announcement. We usually do it in November, I think, something like that. So she's phenomenal. Get to know Ms. Hewitt. And with that, let me invite Dawn Hewitt to the podium.
Thank you, thank you, thank you. What an introduction. He doesn't know that most of you know me already. Well, I'm actually just here today to introduce our phenomenal guest speaker today. I've had the privilege of knowing this person. We were talking about it for more than 20 years. So Dr. Ramirez is the Dr. Amy and Jane Elster Professor of Connective Tissue Diseases in the Department of Pharmacology and Systems Therapeutic of the Mount Sinai School of Medicine in New York City. He graduated in 1969 from the University of Palermo, Italy, with a doctoral degree in the biological science. He's a geneticist. After his postdoc training in Columbia University, in 79, he joined the faculty of Rutgers Medical School, and he is currently with the Mount Sinai School of Medicine. So please put your hands together and give that great York welcome to Dr. Francisco Ramirez. I want to hear it.
Thank you. First and foremost, I'm going to apologize for the accent. Okay? I'm actually not anymore on Mount Sinai. As of February 1st, I retired. Woo! Go for it. And when Dawn invited me for this event and asked me to give this talk, she told me, just give a talk about what you have done, why you end up in research, and what you are going to do. And having retired, that gave me a different perspective because I'm looking from the past, checking out how did I get there, whatever there is. And so I struggle a little bit how to prepare this talk. I also understand that the students here are of different interest in different disciplines. So how do you maintain a sort of interest from all of you before you start to look at your phone and ignore me completely, which is the story of my life. So what I tried to do was to give you how I end up doing what I did, the reason why I did it, and in so doing, I'm going to show you, at least for my discipline, for biology, for biomedicine, how this system, how this discipline has evolved during time, and show you how my career parallels the change that have occurred in biology and how I use these changes to advance questions that I hope you will see are interesting, at least. I would say also important from a biomedical point of view. So to begin, I would like to remind all of you that today, as it happens, is a National Teacher Appreciation Day. Yeah, for all the teachers. I'm not anymore. So yeah, go for it. So the title, as I say, reflects the kind of talk I'm going to give you. And to begin with, let me emphasize that my training, my undergraduate training, was in Italy. So it's very different from yours. There are a number of similarities, but a number of differences. So let's go back. Unfortunately, Dawn revealed my age. So let's go back to when I was a student and I was facing the question that you probably are dealing with today. What am I going to do when I grow up? Right? I just finished my undergraduate courses, everything. I graduated in liberal arts, so I didn't have a real background in science. And I knew a few things. I didn't know really what I wanted to do. And I knew that I didn't want a 9-to-5 job. I knew that I wanted a job that was interesting, and because I'm a curious person, that will stimulate me. And I knew for a fact that my choice was going to be driven by two basic concepts: no math and no blood. And you will see that both of those things came back to haunt me. Okay, so if you do that, if you eliminate math and if you eliminate blood and you're still interested in the general idea of doing something that is good for humanity, then you end up going to biological sciences. And there, as Dawn told you, I specialize, if you will, in genetics and molecular biology. Those were, at that time, disciplines that, well, genetics was well established, but was the classical one. I was working on Drosophila, that these small flies that you killed all the time. For me, those were my thesis. And it was very descriptive. And molecular biology was really primitive. So I just got my thesis. I graduated in 1969, and congratulations, that is me in 1969. And the question then was, what you do now? You can become a teacher, you can go into industry, you can do this, you can do that. I really wanted something more, and the opportunity came for a fellowship to come to the United States. Okay, so I didn't speak English. My background was, okay, but wasn't that good. And I was young, so I said, well, what the heck? This is the age that you are supposed to make mistakes. So let's try this one. And I arrived at Columbia Presbyterian, where I discovered that half of the population didn't speak English either. And given my name, I realized that I had a lot of cousins in the neighborhood. So I worked there at Columbia, but Columbia was an important thing for me because I was very lucky. I came to a time in which biology and genetics were getting together in trying to understand human diseases. And the disease that they were working at Columbia were the thalassemias. Thalassemias is a general name that means thalassos is for Greeks is the sea, anemia of the sea, anemia of the Mediterranean area, but actually is well spread throughout the world. Down in Africa, particularly in Eastern Africa, and the Caribbean and Asia. It's a genetic disorder that affects the hemoglobins and impair the function of the hemoglobin with the consequence of causing anemia. And there are three forms of the disease. At the time, they were recognized three. A major, those are clinical definition, a major, an intermediate, and a minor. And a major is lethal. And the only thing that at that time that could be done was transfusion. The problem is with transfusion that you start to accumulate iron, and iron accumulation has severe consequences for organ function, particularly the heart, the spleen, and the liver. So at that point, in those days, the question was, can we understand this disease at the molecular level? We know that it's a single gene, and there is a single protein as a consequence that is altered. But can we understand and define, sort of stratify the disease at the molecular level so that we can have an understanding of major, intermediate, and minor? What does it mean that? And the luck was, as I said, I was in a lab that was one of the very first to do gene cloning and DNA sequencing. Those were, you take it for granted, but those days, those were really new things that were coming up. So my training really elevated substantially by learning those things. And I gave my little contribution as a postdoc. As a matter of fact, I was the longest postdoc that I ever heard. I was there for 7 years. And the study of thalassemia in general brought three concepts. We are in the early 70s at this point in time, to put the things in perspective. The fact that we could do genotype-phenotype correlations. I told you, if you clone the gene, you find out the mutation, then from the nature of the mutation, you do an association with the clinical phenotype and figure out how to then do a DNA-based prenatal diagnosis and therefore deal at least with the cases of the major thalassemia. And the other concept that came out is if I have a gene and I know that what is the mutation, can I fix it? So the concept of gene therapy came about in 1978. 1978, and yet it's only this year that they have started the first gene therapy in thalassemia and sickle cell anemia. I say that because you have to keep always in mind that things, the idea that you may have, I want to do this, I do this because I can cure that. It takes a long time to get there. So at the time, I was sort of like, they have discovered everything. I can't do anything new. And I was so wrong, obviously. Now, 1970, late 1970, I had, at this point, after 7 years, I had to decide, do I go back to Italy or do I stay here in the United States? And I say, well, I stay here without knowing what does it mean to have an academic career. I had no idea that you have to write grants. I had no idea how to get money. I had no idea that you have to... It was just a jump in the dark, but I did it. And I took a job in Rutgers Medical School at the Piscataway campus, which I understand the Piscataway is an old Indian name that means there are no good restaurants in New Jersey. And there I went there because I had these tools that I had learned, cloning, sequencing, human genetics, and all this other stuff. And they were interested in a group of disorders, which I will show you in a moment. And they had really a group of biochemists that were working on protein chemistry and the interest in resolving this, the cause of these diseases. So what did I do? I took the startup funds, I put together a small group and seriously went on after research. New Jersey is a very lonesome place. These are the disease that I studied from there on. And it's a group of disorders, heritable disease of the connective tissue, which are extremely rare. Some are more frequent than others, but in general, they are considered rare disorders. However, there are about 300. If you take them all together, they are quite frequent. And they affect all sorts of tissues, obviously. You can tell them just by looking at these pictures that one of the tissues that is mostly affected is the skeletal system, the musculoskeletal system, and also the skin. So the phenotype are there, but some others are much more complex because affect other tissues. The complexity of the phenotypes reflects the complexity of the molecule, the protein, that constitute the element of the connective tissue. And without putting you to sleep, because I do, speaking of extracellular matrix, the extracellular matrix is essentially a building block of our tissue. And there are essentially 3 groups of molecules there. The architectural components, molecules that form this big structure that you can even see. And those are collagen assembly and elastic assembly. Adaptor molecules that adapted these molecules so that each tissue has a different architecture, 3-dimensional architecture, so that the biomechanical property of the tissue can be different from one tissue to another. And lastly, ACM-related enzymes that are required to modify this protein so that you can build it up. So essentially, it's like a Lego system in which you have different colors and different things. And with the same component, you can make different shapes, different forms, different things. And here is a EM picture, electron microscopy picture, showing the inside and the outside of the cell. If you look at that level, you recognize if I didn't put that dotted red line and not written there inside, outside, you could not tell where the cell ends and the outside starts. And that is important because essentially what it tells you is that the division that we say that we have between cell and tissues, a connective tissue, is an artificial one. The cell makes the tissue, and the tissue tells the cells where they are so they can continue to behave in the correct way. When the tissue is degraded, when you have, for example, a disease that destroys the connective tissue, the cell don't sense anymore what is around. And the sensing is a structural sensing, if you will, because as you can see here, okay, forget it. As you can see here, the connection between the inside and the outside of the cells lead to a sort of continuum, which is not only structural but only functional. Why I say that? Let me show you an example. So let me take this disease. It's called epidermolysis bullosa. Epidermolysis bullosa means that people affected, most children affected with this disease, have a skin that tears just at the minimum bruise. And that can involve the entire body or just area of it. If you look at them, the approach of isolating genes and looking at them for mutation and establishing genotype-phenotype correlation does also another thing. You look at the epidermolysis bullosa, there are 3 different types that you can see going from the simplex to the junctional to the dystrophic, being one, the dystrophic, the last one, the most severe. And you can see the list of mutation, K14, K5, ETG, COL7. And you can see also in this slide, in this scheme, where the mutation affects the continuity between the inside of the cell and the outside of the cells, in addition to the membrane that maintains that connection. So by identifying these genes, not only we can start to understand how the disease evolves because where the mutation and the defect occurs, but also you can start to lay down what is the molecular structure of our skin. You have the cells, and we know what are the cells, the keratinocytes, and then underneath the fibroblasts and so on. What we didn't know before was how molecules that biochemically you could separate just by extraction were actually organized. And that is something the cells did. But the maintenance of this structure was absolutely necessary for the cell to survive, because when that structure is eliminated, you have the consequence that you have seen. Okay, so here, my first luck. Here is a student, Brendan Lee, who comes also from CUNY, an MD PhD. I was lucky to have it in my lab, who changed the direction of my lab. At this point, I was at Downstate Medical School in Brooklyn. I went around a lot. You know, being Sicilian, that is a good thing. You never want to be too long in the same place. So what Brendan did, he did a thesis on describing for the first time what is the mutation in a form of dwarfism, disproportionate dwarfism. And he had to go back to the clinics because he had completed the PhD phase of his MD PhD. We had the opportunity to go after the gene that caused this disease, Marfan syndrome. I will go back to Marfan syndrome, why it was important and why we made the Nature cover with that discovery. What he did, what he decided to do, say, okay, I'm going to take the risk of delaying going back to medical school to go after this gene, and he did it in 6 months. 6 months later, he had the gene. We ended up in Nature, and you can see that for whatever reason, they have Lincoln there. Because they thought that Lincoln had Marfan syndrome. It didn't. These things of figuring out, it was good for us because it got us, everybody said, oh, you got the Lincoln gene. No. But it was a good thing because it really allowed us to develop a research program that had at this point an end point. It was not anymore descriptive. I say that genotype, phenotype correlation were important, identifying where the mutation causes and so on, but it was still descriptive. So let me describe what Marfan syndrome is. So it's actually a connective tissue that is the most common of all. It's 1 out of 5,000 people are affected by. Has no preference in terms of ethnicity or gender. the severity ranges from neonatal lethal to asymptomatic. And the most interesting thing is that the same mutation in a family can have a different expression. That is, in the same family, a person may die, I will tell you of what, because of Marfan early on in life, and somebody else in the same family who is affected may not display any expression of the disease. So the disease is variable both across different families and within the same family, and that is almost unique. From a biological point of view, if you think about how can a single mutation have, of this protein, have such a variable expression in mutation, what it tells you is that there are what geneticists call modifiers. What does it mean, modifiers? Each of us is different genetically, even though we are similar. A minimal difference may change the expression of a gene in the context of your genome. So modifiers means that that particular protein, that extracellular matrix protein, interacts with many other stuff, many different things. So let's go on the clinical manifestation. Clinical manifestation is, I say, is multi-system. They affect many organs. It's very difficult to do the diagnosis except for these three hallmarks, if you will. The ocular system, patients have, 80% of the patients have ectopia lentis, which means a dislocation of the lens. The skeletal system is very interesting. They have longitudinal bone overgrowth. For these people usually are very tall, very long spidery fingers, and the growth is with the limbs are the one that they grow more than the rest of the trunk, therefore have this classical appearance. But the problem with the Marfan syndrome is the cardiovascular system, because people that are not diagnosed with Marfan syndrome on time, or we are not under, at the time, any kind of medical management, they are on average dying around 30 years of age of dissecting aortic aneurysm. That was a ticking bomb, that a predictor that affected the survival of these individuals. What the protein is that causes the disease is called fibrillin, and is a component of the elastic fiber. So the elastic fiber, as the name implies, are structures, architectural structure that provides the tissue with elasticity, which is the ability to stretch and recoil, which is extremely important for the cardiovascular system, but is also important for breathing, diaphragm, for speaking, all the system, pulmonary system, and for all other, for joints, the kidneys, you name it, every single tissue has the presence of these molecules. So let's go in the aorta. So this is the aorta. The aortic root is where it comes from the heart. And the aortic root at the initial part of the ascending aorta has a very important biomechanical function, because it's the site where the blood pumped from the heart goes in, and the aorta expands, absorbs these forces, and transforms these forces in a positive way for the blood to continue the circulation. The fibrillin and the microfibrils are exactly where one would expect in the aorta, in the tunica media, which is the internal part of the aorta, where are these smooth muscle cells that contract and relax, allowing the system to function between the systoles and the diastoles. When you have, when in the Marfan syndrome, what you have is a progressive dilation of the root of the aorta, which can reach 10 centimeters of this. And eventually, that aneurysm will dissect, and the blood will enter the wall of the aorta, and eventually it may dissect throughout the wall, and people will die of dissecting aneurysm. When you look at the histopathology, on the left in A is a view of the elastic fibers and in between the cells in a normal aorta. And B, on the other side, is the one of Marfan. As you can see, the Marfan one is completely degraded. What can be done for the disease? The management used to be lifestyle changes, doing low impact sports, regular echocardiography once you are diagnosed with the disease to see how the growth occurs, use drugs that will decrease the antihypertensive to decrease the injury from the blood flow. And last, which was demonstrated to be the most important one, prophylactic surgery, which is described on the panel below, in which the entire segment of the aorta is replaced by drug tissue, an artificial tissue. It can involve also the aortic root. And patients have now, with the Marfan, now survive in their late 50s and 60s because of this. However, the quality of life is very much impaired because, as you can imagine, an artificial tissue doesn't have the same elastic property of the regular tissue. So eventually what can happen with these patients is once they have their aorta replaced, they will start to dissect distally from it. So you keep repairing. So it's still a disease that that has some problem in being diagnosed, but also has affected substantially as I say, the quality of life of people affected by. Okay, so here we go again. Okay. To move on in trying to understand what to do with the Marfan syndrome, we needed to have an animal model system to study the progression of the disease so that we could figure out a way to intervene to mitigate the progression of the aneurysm. To have a disease model, once again, lucky, in the 90s, came out all the technology of mouse genetic engineering. And one of the people that was really one of the pioneers in doing so was Rudolf Jenisch. And we had, again, the luck of interesting Rudy in collaborating with us so that we could generate a mouse model for Marfan syndrome, which we did. And the mouse had all the features of the human disease. The skeletal, you can see that there is hunched, and if you measure the bones, they are longer than normal and so on. But most importantly, the mouse on the right side is a survival curve. On the blue, you see mice, wild-type mice that live a nice life after 6 months, whatever. And on the black one, you see how the survival curve of our mouse is. And as you can see, they all die before 6 months of age, which replicates from 30 days to 150 the spectrum, the clinical spectrum of the human disease. So the model functioned for us for what we needed, which was replicating the human disease. Now, the first finding that we had was, I think, the most important one, and the one we rely for all the future research. The thought at that time was that since we're dealing with the structural molecule, the the disease is probably due to the fact that the elastic system is never formed in the correct way. And once the people are born and subjected to higher blood pressure and higher insult from the blood secretion and so on, this secondary effect of the hemodynamic stress caused the disease. If that were true, we should have evidence from the mice that there is altered structure of the elastic fibers at birth. What instead we observed was that the mice had the aneurysm. The top is a wild-type mouse heart, and you see the aorta coming out. And lower one, there is the aorta that is larger, so that is an aneurysm. When you look at the section, histological section, this is a cross-section look from up, you can see a dissection occurring in the mouse, is where the black arrow is, and there is the infiltration of the blood that is breaking into the wall of the aorta and eventually will go out. But if you look very carefully, you see all that black shade there all around. Those are the elastic fibers. So the elastic fibers, they look, at least morphologically, okay. So the mutation in fibrillin do not alter elastogenesis, the formation of the tissue. What they alter is the ability of that tissue to sustain the hemodynamic stress. That is important because in the first case, the original one, therapeutic intervention could be done postnatally, but they were limited by the fact that the tissue was destroyed anyway. In the second case, what we observed is that the secondary effect of the hemodynamic stress could move the therapeutic intervention before the tissue become degraded as a result of the hemodynamic stress. With that in mind, we and many other people started to look for what kind of molecules are affected during the hemodynamic stress, and can we pharmacologically target this event? And so everybody started to look at molecules in what we call the candidate pathway. So you have, you look at the issue, you see that there is fibrosis, accumulation of collagen, and you go for the TGF-beta pathway. Sorry for using the jargon, but that is a pathway that regulates fibrosis. That is informative, but doesn't do anything to put in context what kind of molecules are involved. In more recent time, we have changed the approach by going into a system biology. And here is, remember, I say mat, no blood. I told you about thalassemia, that was the blood. And now comes the math with the system biology. So system biology, and you guys know more than I do, is a computational and mathematical analysis and modeling of complex interaction within biological systems. So rather than looking at the relationship, the classical relationship, gene, protein, phenotype, we are looking at a phenotype as being the consequence of an alteration of a network of interaction that occur among different genes, right? So it's a genome interaction. That is a molecular networks. If you look at the higher level, you can look at the cellular networks, the signals that cells from one tissue to another communicate as a result of an alteration in one of them. Or you can go at even higher level at the organ network. Now, with my background in no math, obviously I couldn't take this one, but again, luck was very good for me because as it happens at that time, I was at Mount Sinai. And at Mount Sinai, the person who had recruited me, and we were next door to each other, is Ravi Iyengar, who is the head of the Mount Sinai Institute for System Biomedicine. So now we had the opportunity to use computational biology and molecular genetics together to try to solve the question of giving a more detailed stratification of what was going on. And just because, again, I'm trying to keep it to a level that we can all, everybody can follow, is I'm going to present you two strategies that we devised with Ravi, actually Ravi devised, that are related on rely on computational biology to ask the question, can I find out unbiased way to approach this disease? So the first one is the so-called pathway discovery. Essentially, we go back to the basic. Genes are activated by transcription factors, proteins that bind on the DNA on specific sequence. So there is a specificity there. These transcription factors are activated by other protein enzymes that are called protein kinase. And there are a number of drugs that can inhibit this protein kinase. So theoretically, from the expression of the genome, we can go back and identify the protein kinase. Now, that is simple to say if you are looking at one gene and one product. The issue is that when you look in total at the total genome, the genes that are affected are many, many, many more. And you can see a volcano plot on the right side that shows all the genes that are affected, either upregulated or downregulated. Upregulated are in blue and downregulated are in orange in the tissue of a Marfan patient, in the aorta of a Marfan patient. So there are 981 statistically relevant genes that pass the threshold. The gray one is genes that are overexpressed or underexpressed, but they don't reach that threshold. And 412 on the other side. So you really need robust computational tools to sort out from this one what kind of transcription factor may work on them. And the way it's done in computational terms is that the binding of this transcription factor occurs on consensus sequence. So you can derive from taking all these genes which one are the consensus sequence in the DNA that are mostly used, right? By doing that, you essentially give a tag to the transcription factor. From that, you can then go, still using the computational tools, to the interactome, which is essentially an atlas that has all the interaction between the protein kinase and transcription factors. Now, it's not a perfect thing because it's based on literature data. And as you know, as a computational biologist and so on, if junk goes in, junk comes out. So there is a limitation. So that all these experiments eventually have to be validated in vivo. When you do that, so the scheme then is to look at genes that are upregulated in the aorta of Marfan patients and our Marfan mouse, right? So you get the generation of those things. In each of this group, you identify your transcription factor, and you then look in each of the group which one are the protein kinase that are the most likely to be the highly expressed, be involved. And when you do that, the mouse protein kinase and the patient protein kinase came with a high score for the same protein, which is that IPK2, which is a stress-related protein kinase that is activated as a result of stress and activates then pathways that are to deal with the stress, being that hemodynamic stress or other form of stress. Now you have to do the validation. So this was Ravi work, then it came back to us to look at the validation, and you do a genetic experiment on the Marfan mice or a pharmacological experiment. In the genetic experiment, you create mice that don't have, Marfan mice that they don't have the IPK2 gene. And in the pharmacological experiment, you use a general inhibitor of this family of protein kinase to see how the mouse survival is affected. And as you can see, the red one is the mouse control, the Marfan mouse without any treatment or genetic manipulation. And the blue one is the mouse that is either without IPK2 or treated with the drug. And as you can see, they still die of dissecting aneurysm, but the disease is delayed, which is a good thing. The delay is associated with a reduction or a normalization of the aneurysm. So the aneurysm disappears or is late in appearing. That is more appropriate. And the degradation of the tissue, which you see in the histopathology on the right side, is normalized in the one that is without the gene and sort of better in the one with the kinase. And the difference tells you that the kinase, unfortunately, is targeting other things. So that it really is the elimination of that gene that has the major effect. Two things. First of all, is not a panacea. It doesn't cure the disease. And number two, it tells us that the dilation and the degradation are two different processes. They are not one after another. They are independent processes that show the stochastic nature of this disease. So, to eliminate the problem that at this point we have identified a pathway, which is good, but then we don't have a drug, Ravi decided to go the other way around. Why don't we look at target drugs that are FDA approved and see if they work to treat this disease? And this is called drug repurposing, and it's based on the fact that we have available what is called the connectivity map, which was an experiment done for many years at the Broad Institute, in which they have treated the cell culture with different drugs. And they have done data analysis of total genome in all the different drugs, with all the different drugs. So they know if you take aspirin, what kind of gene are upregulated or downregulated, right? So then, this is the construction of the experiment. Once again, you take mice and human tissue, you look at all the tissues, all the genes that are up or downregulated in each one of them, and you translate this gene, this protein, in cellular processes. That is contractility, proliferation, you name it. And then you identify what are the cellular processes that are common between the human and the mouses, a little bit like I showed you before. The difference is that at that point, you go to the connectivity map and you ask the question, are there drugs known to counter the effect that we are seeing? That is, if you have an upregulated genes, do we have a drug that we know does downregulate that cellular process and vice versa? So when you do that, and when we did that, we realized that both the mice and the patients share a number of genes that are associated with contractility, which is not surprising because contractility is what is the base of the aneurysm to begin with. Based on that, we looked at the connectivity map and scored for drugs that were used for other things that may revert, that is, upregulate those genes. And the drug that came out to be very at the high were fludrocortisone and baclofen. Fludrocortisone was excluded because it causes hypersensitivity, and so it would be contrary to the use. But baclofen was one that looked good. Baclofen is a drug that is used for muscle spasm. So it's very common drugs. Has the advantage that has been in the market for so long that we know what are the side effects. So we don't have to go through the clinical trials and all this other stuff. And when we did the experiment by using a dosing that is equivalent to the human dosing in mice, we found again that the mouse survived longer, that the aneurysm was decreased but not eliminated. You can appreciate the difference with the others. And its tissue was sort of restored. So this is an approach that is very promising and that many people are looking into it in order to identify pathway and drugs that could be used for the intervention. Now, these are the last two slides. This is sort of summary slide. So we have this disease process of aneurysm progressing, dilatation progressing to dissecting aneurysm. And I told you that we have tried in many different ways to approach it, and that the computational approach is the one that looks the most promising for the future. Particularly also combined with the nanotechnology and the search for small molecules that can interfere with some of the function of target genes. But the future, which I'm not going to be involved in, thank God, is how the disease also progresses, how this communication occurs within the tissue, what we will call the tissue networks. The intima, the media, and the adventitia, the three layers of the aorta, endothelial cells, the smooth muscle cells, and fibroblasts, they communicate with each other. And this communication are reciprocal. So what we have to solve now is tissue network. And tissue network means all these genes that we find that are high, that are candidate genes for association with the disease, that are either upregulated or downregulated, they have to be distributed within the tissue. Are there cells that are particularly susceptible? So do exist in the smooth muscle cells a subpopulation of cells that respond more to the damage of the hemodynamic stress and therefore starts to communicate with other cells. And the single cell RNA sequencing is a new technology that is very much the one that could identify subpopulation of cells that may be important for disease progression. And spatial transcriptomics, which is again another high resolution kind of approach, is the one that can then distribute this pattern of expression in different sections and pattern of the aorta. And then I put a question mark there, because like it happened before, throughout the history of this connective tissue disease, we don't know what is the next technical breakthrough that can help us solving other biological problems. So I'll get to you to my closing remarks, the 7 principles that I learned in this sort of journey that I had starting when I was asking myself what I will do when I grow up, to now that I have retired and I'm not much to do around, okay? So the 7 principles. So you are students, and this is something that I always told my students when I recruited or when I was mentoring and so on. So the first and foremost is follow your instinct. That doesn't mean that you have just to do everything that you feel must be done. Follow your instinct, but be informed about what you're going to do. So you're at the point in which you have to make some decision, right? Inform yourself, talk to people that have gone through the same thing, and see how much. But then at the end, you have to go for what you really like to do. In my case, when I finally moved into the lab, I discovered, no, this is the place I wanted to be. This is the sandbox. I can play. So that is the first one. The second one, and this is the most important, I would say, is love what you do. Because if you're going to go into science, no matter if you do basic science or applied science, if you're in academia, or if you are in industry, if you're teaching, if you are whatever you're going to do, if you don't love it, you will hate it. It's really a binary choice. The third one is don't be afraid to take risk. Many times we are very risk adverse and we try to be in our comfort zone. Comfort zone is boring. I can assure you. So risks have to be taken. And you're young. I always say it, and I said it before, one of the must at your age is to make mistakes, because that's the only way you can learn. And that is how we do science. 99% of the experiments don't work. But you need those negative results to understand when they work. The next one is capitalize on opportunities. If comes across, don't be afraid to take an opportunity, but make sure that that is what will take you to the next level. Don't take the opportunities to repeat yourself. Move on. The third one is don't be a one-trick pony. What I mean by that is that often we learn something, again, we go back to the comfort zone, and we stick with that because we are good at it. The problem is, as I show you, as we went from the early days of human molecular genetics to today, where we have computational biology, soon there will be AI and all this other stuff, if you stayed only with what you knew, you will never get to what is moving on for the future. 6, always work with people smarter than yourself. It's not about you, it's about the data. And if you look at people that you can show off, it doesn't work. And the last one, don't stop learning. Don't be sure that you know what is going on. There is always another corner to be going around. So those are the principle of the things I've learned. My last consideration is what I had in the title. What do you do? Would you do it again? And the answer is yes. I will do exactly all these things. And I made a lot of mistakes. I didn't tell you all this stuff that was. But it's yes. And the yes is modified by one thing and one thing only. There is one thing that really makes being in research something precious. And is the fact, the excitement of the discovery. The first time you see a result that finally tells you yes or no about one idea and so on, and you are the only one that sees for the first time. I remember when I was a postdoc, the first time I saw a result we always wanted to see. That is priceless. And that is worth all this sweating that we have to do to keep going and to research and to modify and to go on and on and on. So I stop here. I thank you for your attention and good luck with your future. Thank you.
So if anybody has any questions for Dr. Ramirez, please go ahead.
Yeah.
So the nature cover showed Lincoln and the association with Marfan syndrome. I also remember from a human genetics textbook I used something about Toulouse-Lautrec and what he may have inherited.
Right.
So could you tell us what they actually did that?
Right. So Toulouse-Lautrec had a different disease. It is a disease that was discovered actually by a colleague of mine at Mount Sinai, Bruce Gell. It's a disease that that affects the skeletal system growth and so on. So it's a different disease. And this bona fide, we know what caused it and so on. Lincoln, we know that it didn't have a Marfan. The reason why they thought that the Marfan because it was very tall and so on, and very skinny and that posture. But they went after we published that paper, they allowed people to go after some bone that from Lincoln that got the DNA and they checked it and so on. But actually, from a genetic point of view, you could have told the same thing because none of the people associated with the Lincoln family had any of the manifestations. As a matter of fact, there was a moment in which they thought that people said that Osama bin Laden had the Marfan because he was very tall, extremely tall, very skinny. Again, there is a manifestation. But there was clearly a mistake because this is a dominant disease. And none of the other people in his family, many children, had any of these manifestations, was not. There are other people that are suspected to have had the Marfan. One is the Rachmaninoff, because of the long fingers and the fact that if you know the piano concerto, they are very hard. Same thing with Paganini with the violin. One person that had the Marfan syndrome and unfortunately died of was the author of the play Rent, Larson. He died of dissecting aneurysm in Greenwich Village the night that Rent moved from off-Broadway to Broadway. And he was misdiagnosed. They thought he had heartburns and was sent back home and he died there. And you couldn't tell that he had the Marfan because he had the normal stature. He had glasses, but you know, we all wear glasses. Only if you had the family history or, and again, there are cases that are spontaneous mutation, or if you had echocardiography, they could have told him because probably his heart, its aorta was already 10 centimeters in size. Okay? Nothing? Okay. Oh. Yeah, it's very easy. Unemployment. I swear. That is why I always say, don't overplan in your life because life always shows you. So yeah, unemployment. I had the techniques. I needed somebody who said, you know, you could use this technique for this question we have. And then I appropriated the question and move on, like we all do. Yeah. Okay. Okay. Oh, no, I mean, there are many other drugs that have been tested. This was just for the sake of argument of this particular case. Just I wanted to show you a protocol that can be used in general for any disease, actually. Okay. No, so one of the, okay. When we first look at the disease, okay, in the mouse, we realized that there was something going on with probably with the involvement, and now I'm going to go specific, okay, with involvement of growth factors that is extremely important for matrix modeling and remodeling, which is TGF-beta, okay? And we figured out that we could target the TGF-beta by using an antihypertensive drugs that has been used in other diseases such as scleroderma, because it has also an effect on fibrosis. And that drug is losartan. And that losartan drug was used in the mice, and the mice were rescued and all this other stuff. And that was then moved from a preclinical to a clinical, and a clinical study was done in patients. Unfortunately, the drug didn't work in patients because a posteriori we discovered that you have to intervene at a certain time, which we did in mice because we can do it. But when you deal with patients, you are dealing with a mixed population. So you have children, you have adults, you have teenagers, and so on. And so you are hitting a different time in the progression of the disease. I try to show you that the progression of the disease is a cumulative effect in which many different pathways come in. So you can't do that. And certainly you cannot do the same experiment in humans. So the thinking now is, from what we have learned on the semi-efficacy of these drugs, is to try to understand where and when these drugs act so that we can start to use multiple different drugs, many different drugs for the same patient. So a treatment that is not only one drug, but a polydrug. Yes? I can't hear, sorry. Yeah, that's okay.
You mentioned that it's the dominant drug.
Yeah.
Would it be possible to just like this for like one of the
Yeah.
sweet spots?
Yeah. Yeah. So the question is, can we use genetic approaches like CRISPR to modify the gene in people, like we are doing now with sickle cell anemia? That is what we are doing. Tell you why not. I mean, I don't want to say no, you never say no. It's tough. So why we did it in thalassemia and sickle cell anemia? The issue is a very simple one, is the accessibility to the tissue. Blood is made by bone marrow. You can take out the bone marrow from a patient, you can change the gene, and you can put it back. You cannot do that with a gene that is expressed everywhere. That is the problem. So gene therapy is something so that is why I was mentioning also nanotechnology now is something that is coming out, because it's an issue of delivery. If you could figure out a way to deliver it to the aorta, you don't care if you have a skeletal problem, you don't care if you have ectopia lentis. What you want to stop is here. So it should be targeted. But then you have to understand what to target. Should you target the endothelial cells? Should you target the smooth muscle cells? Should you target the fibrous? Should you target all of them? Those are, in case you are interested, those are the challenge for you guys to address. Okay, can we go home?
They're not going home yet.
Okay. Thank you again.
I know Dr. Ramirez started his talk by telling you when he came here, he didn't realize he had to beg for money and get money. Well, he was very good at it because he was very, very funded, his labs, by the National Institutes of Health. But the one thing, I met him 25 years ago at the Hospital for Special Surgery. And my office and his office were on the same floor, and so was his lab. The thing that impressed me the most about him was how diverse his lab was, and how, as the director of the lab, he was always hands-on with his labs and with his students who have gone on, and he'd encourage to grow and let them go and continue. So the 5 years I was with him, just to see that. And he also impressed my daughter, who used to come to visit, that she liked going to his lab because it was so nice and friendly, and he was so nice and friendly. So that's what he said about enjoying what you do and the people that you work with, because everybody enjoyed working with him. You worked hard, and his lab was always fun, and it was always like a family. So we would like to thank you again, Dr. Ramirez, for all that you've done and your contribution to science.
All right, so what we're gonna do, since some of the students have not been able to present earlier for the poster, we're gonna go back to the poster area, and your poster, we mingle again, and while we're doing that, we're gonna award the certificates. And then we'll close, basically we'll close and okay? So we'll award the ceremony, I mean the certificates, and title ready, basically, as your man in the poster. And thank you for a good day. I hope you enjoyed it. Yeah.